Hi, I'm Dr. Huckaba, and the work in my laboratory is focused on a series of diseases that we refer to as neurodegenerative disease. And you've probably heard of a few of these before, but these are diseases where the nervous system forms normally. So these aren't developmental diseases. A normal nervous system starts to break down over time. And we're focused on two of them specifically, Alzheimer's disease, which you've likely heard of. We're working with Dr. Sridhar in the chemistry department. Her laboratory is generating some novel compounds that are potential therapeutics for Alzheimer's, and my laboratory is doing the biological testing to see how effective they are. The second disease we're interested in is called hereditary spastic paraplegia. That's maybe one that you haven't heard of before, but we're trying to understand how mutations in a protein called kinesin ultimately lead to this disease. And the tools that we use in my laboratory are essentially twofold. We utilize biochemistry and cell biology. And it's not like one project is using biochemistry and the other is using cell biology. What we try to do is sort of build synergy between these two systems, and I'll show you an example of that. So, for example, here on the left, you can see this cartoon of a kinesin. That's this protein that's walking along. And the substrate that it's walking along is called a microtubule. This happens in cells. This is how things get moved around in cells. And so what we can do biochemically is we can purify the kinesin, the thing that's walking, and the microtubule, the thing that it's walking along. And that's what you're seeing in this movie in the middle. These are purified components, where in green you're seeing a kinesin walking along a red microtubule, and this is a high-resolution microscopy technique that we use in order to visualize single molecules of kinesin walking along microtubules. But then we can also study the cell biology of that. So these little bright dots that you're seeing, this is a cell expressing a fluorescently labeled kinesin, and you can see it walking along. It's a pretty quick movie. Let me start this again. Sort of walking along the microtubule array inside the cell. So what we're learning from the biochemistry is also we're using the cell biology in order to better understand what's going on. And this is what I mean by sort of the synergy between these two major techniques in our lab. And so you may be asking, well, if you join the lab, what will you be working on? And the answer is all of it. I don't, train students just to do one thing. I think it's important, particularly as an undergraduate, that you're broadly trained in techniques, and techniques that aren't necessarily specific to my laboratory, but techniques that you will use regardless of what field you go into as part of your larger biomedical research career. So for example, things like cell culture, PCR-based cloning, high-resolution microscopy, protein expression purification, in vitro reconstitution, sort of rebuilding biological activity out of purified components, and then some basic techniques. Those aren't just used in the research around Alzheimer's disease, for example. If you went and worked on cancer later or vascular disease, you would be using these same techniques. And so as an undergraduate, I think it's really important that I broadly train my students in such a way that you're prepared for your later career, regardless of what direction you decide to go with that. And just as importantly, I think the other thing that I really stress in the laboratory is getting my students to think like scientists. I mean, you've heard discussion of the scientific method where you have this hypothesis and then you design experiments to test your hypothesis and you take that data to refine the hypothesis based on the outcomes of those experiments. Well, in the lab, I am constantly asking questions of students. What is it that you're testing? What's the hypothesis? How will the outcomes of this experiment? What if it turns out this way? What does that mean for our overall hypothesis? Because what you're learning to do in the laboratory is not just techniques, those are important, but ultimately learning to think like a scientist. And that's something that I think is really critical skill set that you pick up. Unfortunately, I don't have currently any students in the laboratory to talk to you about the lab in preparation for the sabbatical that I'm on now, and also with a little bit of help from COVID. My laboratory is empty of students, but I have a picture here of these are all former Build students in the laboratory who've moved on to life after Xavier. They've all in graduate programs, either working on PhDs or have gone on to medical school or dental school, and I would be more than happy to put you in touch with any of them if you'd like to hear what life is like in my laboratory. But I'd also can talk to you one-on-one if you'd like. Although I'm out of the country right now, I'm more than happy to set up a time to Zoom or to email, and we can discuss a little bit more about what's going on in my lab.
All Right. So I'm Mehnaaz Ali, Dr. Ali to you guys. Um, so in our lab, we are kind of working towards the general goal of developing biosensors. Now, especially during the time of this pandemic, we've all been in the first half of this pandemic. We essentially were talking about testing, testing, testing all day long. And essentially, the COVID test, regardless of which kind you're talking about, is an example of a biosensor. Now, how do biosensors work? In general, there are some general things that they require. So they need a molecular recognition entity and something that's going to help you present a signal, right? So that when you have your analyte and your molecular recognition entity sees your analyte, whether it's the COVID antigen, whether it is the flu, whether it is lead contamination in the soil, doesn't matter what it is, right? When your analyte specific molecule recognizes that your antigen or your toxic chemical of interest is present, then what's going to happen is there should be some kind of signal, right? Some kind of change, whether it is digital or a numerical or visual, that tells the researcher or the person on the other side that yes, indeed, there is this molecule that's present. Now, what are some classic biosensors out there that you may have seen? Well, of course, we have the pregnancy test, which is a classic example of a qualitative sensor, basically something that tells you yes or no. Yes, I'm pregnant. No, I'm not pregnant, right? At this point in the game, it's not how pregnant am I, but is it a yes or no? That's essentially it. That's qualitative. And then, of course, there's the quantitative test, like the blood glucose meter. This is something that we're all familiar with. Either you have a family member using it, or you have a friend or somebody that's essentially monitoring their blood glucose. This is a classic example of a quantitative test, something that gives you numbers, a numerical representation of how much of your particular analyte is present in whatever media you're interested in. So what we're trying to do in my lab is essentially build the chemistry that goes into these kinds of instruments, okay? And specifically, we're interested in building, and we are working towards building sensors that either target particular types of cancer that are difficult to detect unless they have progressed, down the line, or we're interested in detecting traumatic brain injury, which is very hard to detect as well. And then most recently, we've started some environmental projects as well, and all kind of using this umbrella of biosensors. In the lab, what we have done is we have, and I'm not necessarily going to go and explain every little bit of what you see here, but these are some two different representations of student-driven publications and kind of general areas that we target in our lab, okay? So either we can have biosensors using aptamers that recognize RNA or DNA, let's say miRNA or a specific DNA that is of relevance to some kind of clinical event, or we can have a sensor that recognizes the presence of a small molecule or a protein. And we have successfully shown proof of concept using our approach for the detection of both miRNAs. In this case, MIR let7i, which is a MIR that's been implicated both in certain cancers as well as in traumatic brain injury, as well as we have successfully shown that we can detect ATP in very, very low concentrations in blood serum. So both of these, all of our work in the lab is essentially carried out by undergraduate students. They are involved, undergraduate students are involved in from the design of the project all the way to helping me write the paper for the projects. And while we go through this process, it may be months, it may be years, and sometimes a student will start a project and hand off the baton to the next student that takes over the project. Because with biological research, sometimes you cannot get it done in a couple of years. So it's a long going, evolving process, and our projects have essentially evolved. Sometimes actually due to the interest of the student themselves, they have helped us direct where our project goes. So for instance, our environmental projects that we have going on was actually designed because a student was very interested in that type of work. Now, what are the types of techniques and instrumentations that students will be exposed to? There, we are a instrument heavy lab. This is true. I'm an analytical chemist, so I like to use every kind of technique out there to help us sort of characterize and move our project forward. There's UV vis, IR spectroscopy, different kinds of mass spec like MALDI-TOF, of course, separation techniques such as HPLC. So a student will essentially be trained by me or a senior student, and frankly, I don't have a senior student right now, so it'll be me, on all of these techniques. And then of course, or on these instrumentations. And while we go through our process, the skills and methodologies that students will be exposed to include protein assays, nucleic acid assays, different organic functionalization techniques, solid phase immobilizations, of course, enzyme kinetics. So this is kind of like a snapshot of what a student will be exposed to and will get trained on in the lab. That was my spiel. And if anyone has any questions that they think of later, just email me, please.
Okay. So for those of you that don't know me, I'm Ian Davenport, and my interests are the evolution of live bearing in chondrichthyan fishes. I'm actually interested very much in general biology of chondrichthyan fishes. For those of you that forget what chondrichthyan fishes are, they are the sharks, the skates, and the rays. And the work we do in my lab is looking at stages in the evolution of live bearing in these fishes. So with the living sharks today, there are species that actually lay eggs outside of the mother, like you see here. And you can actually see the little embryo inside the egg case with the egg. So these are laid outside of the mother and left in the marine environment to develop, which is a pretty harsh place. So they have to hope they survive and hatch and then start contributing to the general population. Or with the living sharks, they go through different kinds of macrotrophy. So ultimately, some sharks give birth to live young via a placental-like attachment. If you're familiar with sharks, then you know that they're apex predators. Therefore, they tend to have relatively low numbers. The young are often born relatively large and advanced. And to do this, the maternal organism has to supply nutrients to the developing embryos. So even with the egg-laying sharks, we tend to see relatively large eggs, with the idea of producing large precocial young. This has been taken to an extreme with placental attachments. So the young attach to the mother's uterus very early on in development, and she will feed them. And we can just make out here a shark that's being born. And it's pretty big, and it's all ready to swim. It's got teeth, everything. And when it breaks away from the placenta, it has a little belly button scar like we do. But in these transitions, there are many steps to go from egg laying to live bearing. And one is the early steps are producing larger and larger eggs to produce a larger and larger baby. You want relatively few offspring, but larger and larger and larger offspring to increase the chances of survival. One of these early stages then, breaking from here, is we keep the eggs inside the female, but make the eggs bigger and bigger and bigger so that the young can be larger. This is a finite system because these eggs can only get to a certain size before they may break. So from this stage, then we see the evolution of different modes of matrotrophy, where the mother will take over, supplying the embryo with nutrients. But my lab looks at this stage and the production of these really large egg cells. And our questions are basically, what's going on in the female reproductive system to produce an egg that can reach 10 centimeters in diameter and weigh several hundred grams? How do we get nutrients to it? And why doesn't it break when it's ovulated and then has to go through the reproductive passages? So what we do in my lab is a lot of histology. We do light microscopy, laser scanning, confocal microscopy, electron microscopy, and we take sections of tissues, put them under the microscope, cut thin sections and stain them. And this is a section through the ovarian wall. The egg or the oocyte is here. This is the zona pellucida. It's an acellular band around the egg. This is the cells of the ovarian wall itself. And what we found going to our eggs here is we were looking at the cytoskeletal architectures to see how they're put together and how they actually stay together. And we see a whole series of tube-like structures in this zona pellucida here. They're not that clear in the light micrograph, so we do all different kinds of microscopy. This green region here is the same region as this. And we discovered all these tube-like structures that are actin-based. And now we're working on the distribution of these phylogenetically and their actual role in producing large egg cells. So it's a novel tweak on the cell cytoskeleton, but instead of being inside the egg, it's actually outside of the egg, as this layer will come with the egg when it's been ovulated. In the lab, we have all the equipment to do light microscopy, frozen sections for confocal microscopy, electron microscopy. We have all the stains and dyes in there. We have everything that we need. I have a series of samples that we can use to get us going. So the fridge is kind of stocked with various samples. But also, I have been going, and students can go if they like, on research cruises. So every summer we have a research cruise that goes out into the Atlantic Ocean. You can go on the RV Oregon 2 and go collect samples if you like. So this is a research cruise, not quite Viking or Carnival. This is more of a hard-working, dirty environment, a bit rough, a bit nasty. So obviously a lot better than most Carnival cruises. This is what we do in my lab.
Dr. Davenport, I have a question. How long are these cruises? How long are you usually gone on the water?
2 to 3 weeks.
Oh, okay. And do you swim with the sharks?
We're not allowed after somebody got eaten the other year.
Ooh.
Yikes.
I'm going to leave now. I've got 12 cats to feed.
Good luck.
They're not all mine. We're cat sitting one.
Thank you.
Bye.
Hi, good evening to you all. Let me start with the type of work that is being done in my lab. What we do here is drug discovery, so it's a part of medicinal chemistry. So it's an interdisciplinary field where it's an interface of chemistry as well as biology. And what I specifically focus on in my lab is the chemistry part and the computational chemistry and the organic chemistry part. So the drug discovery process is a really drawn down process where it generally takes somewhere around 10, 15 to 20 years for a concept idea to come into clinical use. So what you will see is that initially the biologists and the clinicians identify a target that is a kind of either a DNA, RNA, or a protein molecule that is misbehaving in a disease condition. By misbehaving, what I mean is that they are either underexpressed or overexpressed. That is, there are less copies than normal, or there are more copies than normal in a cell of that particular biomolecule, which can be either a protein or an RNA. most of the times it will be the protein that is overexpressed or underexpressed. And these can be the result of DNA mutations or other kinds of signaling molecules like hormones or other small molecules like external cancer-causing agents or environmental agents or some pollutants. So all of these will cause a abnormal expression of the protein. And once we identify that this particular protein is abnormally expressed, that's when the chemists come in. So what we do is we look at the structure of the protein and we identify the way it is acting in the cell, how it is carrying out its function. That is what we look at. And once we get an idea about that, then we try to use agents, chemical agents that can actually target the activity of that particular protein. So once we have that idea, then we start designing molecules that can actually target the protein, bind to the protein and change, either completely inhibits its activity or enhance its activity depending on what we want to do. And by looking at how it is actually acting or how the chemicals are acting on the protein, we may have to tweak the structure of the chemicals to make them better, to make them work on the protein in a better way. So the basic two areas where I'm focusing my research is on refractory and metastatic cancer and neurodegenerative disorders. And what tools do I use? I use computational molecular modeling, organic synthetic methods. I also use bioassay methods to understand how the proteins are behaving in the presence of my agents. All right, so as you can see, I use different methods here. And based on those methods, we find different new molecules that can act as therapeutics. Okay. Sorry for the interruption. I had to take care of a student who is taking an exam. The main thing is a student working in my lab will get exposed to different kinds of things, such as they will get exposed to the different software that are being used for computational studies. And we will do the organic synthesis of the design molecules in the laboratory. So we use many different organic synthetic methods. And we then follow it up with the bioassays. That is, the student will gain experience in the cell culture and cell viability assays. So a student working in my project understands the interplay of the different disciplines when we are looking at medicinal chemistry, especially how therapeutics are being found and how they finally translate into clinical use. They look at the different processes and different stages that a therapeutic goes through. And most of the students get to present their work as poster or oral presentations at national conferences. And they have the possibility of becoming co-authors in a manuscript, depending upon how much of work they have done in the lab. Any questions?
Are there any computer skills we should have prior as far as going into the computational chemistry component of your lab? And as far as your previous research, how much have students been implemented into the research, and how hands-on are you in the lab?
So at present, I have 4 students working in my lab. A couple of them are also doing computational work, and one person is specifically doing only the computational chemistry work. And even during the pandemic, they were able to at least do the computational chemistry work using the software that I made available to them, so that they did not lose any time in the research. And once they were able to start working in the lab, many of them have started working in the lab, performing reactions and carrying out bioassays and things like that.
Okay. So you said you have multiple students in your lab. So are they more like divided out into each component of, like, you say you have one student on computation, other on organic, and another on bioassay and testing?
The person who is solely working on computational chemistry has a different background. She has a bioengineering background, so she is looking at a different aspect of the entire drug discovery system and also drug delivery system. So she is in bioengineering molecules. So she's working solely on computational chemistry and doing energy calculations and all that. But all of the others, they get experience with both computational chemistry as well as organic synthesis work in the lab. And a few of them will be able to do the bioassays too, depending upon their interest.
Okay, thank you.
You're welcome.
Thank you so much. I appreciate it.
Thank you, Dr. DeFreece.
All right, guys, my name is Galina Goloverda, and Dr. Goloverda, of course, you know, and my project is multidisciplinary. I work in close collaboration with Dr. Kolesnichenko, whose background is inorganic and material chemistry, and mine is physical organic and organic chemistry. So, well, the project, like more toward me, is novel linkers for advanced hybrid materials. And by hybrid materials, what I mean is that's a combination of inorganic and organic materials. And first, why is it necessary? You probably heard a lot about nanocrystalline materials, and those can be magnetic. They can have interesting optical properties. Then they can have variety of properties. And this part, like magnetic nanoparticles, it is where Dr. Kolesnichenko is. His background is. He makes very good nanoparticles out of iron oxide and other metals. Some of them are magnetic. But the naked nanoparticle cannot actually do anything. And of course, if we are, you know, looking toward like medicinal aspects, then iron oxide is not soluble in water, as you can understand. Well, so you can't carry the iron oxide inside of the body and have it soluble. Well, what we need to somehow solubilize that iron oxide if we can get a use out of it. And the uses, by the way, are very, very different. You know, there is a variety of uses for nanocrystalline materials. And that varies from medicine, like they can be imaging agents, meaning like, you know, you can track things inside of the body. They can be cell labels, you can label cells with them, or they can be drug delivery machines, agents, stuff like that. But, you know, and that's medicinal part. But you can also have them useful in so-called photovoltaics. That means like solar cells and stuff. So I was going to show like two different aspects, and both of these applications, and that's only a couple of applications. There are many more. They all rely on organic shell that makes it possible, you know. And as I mentioned, for medicinal purposes, we need a shell that would solubilize iron oxide. It will never be soluble like to make real solution, but you can make a colloidal solution out of it. So we are making these linkers right here, which can bind a biomolecule and carry it within the body. Or if it's the different aspect, you can make well, but let me finish with this one because, you know, that will be a little bit different. So for medicinal applications like cell labels, imaging agents, drug delivery agents, you need something that would dissolve that composite. And of course, it's inorganic, organic composite. And we lately, I mean, within a couple of years, we didn't exactly discover this molecule, but we found the molecule that makes like perfect sense as a head for those nanoparticles. Like this head coordinates ions on the nanoparticle, coordinates and holds them, right? And then you can, extend that tail of the molecule to make it bigger or smaller, depending on what you want. And if you look at this picture, you can see like relative molecules to this one. This one is called 2-hydroxy isophthalic acids. If you can see, you have 2 carboxyls in this molecule, right? Or you have carboxyl and hydroxyl in this molecule. And you can also see how solution of iron oxide nanoparticle behaves. And you see how this solution separates, right? So it is not going to stabilize that. You can't really see very clearly here, but solution is turbid here. So stabilization by this molecule is not that great either. But if you have this molecule where you have 2 carboxyls and hydroxyls and a hydroxyl in between, that makes it perfect stabilization. If you look at this solution, it's almost it is transparent, basically. There's no turbidity. So we found this head that can do that. And now in the future projects, we are developing different tails for this molecule. And if you look here, then, as I said, depending on how large that tail should be and what biomolecule we want to bind to it. Or in case of solar cells, well, those nanoparticles will be different. That would be titanium O2. And that is just one electrode that is shown. I hope I'm not talking too much. Okay, 1 more minute, right, Cecily?
Ish.
Ish. Okay, I'll be quick. Well, this is just one electrode. So it is covered by dye sensitizer that, and our tail will take electrons down to this electrode. And the second electron doesn't need a sensitizer. And that's how you basically, roughly, make that solar cell. Well, what methods do we use? A variety of methods. Students in our lab are exposed to inorganic synthesis, organic synthesis, product isolation, purification, nuclear magnetic resonance, IR spectroscopy, so-called zeta sizer measurements, mass spectrometry, most of times it's electrospray, mass spectrometry. And I would finish here. Okay, very good then.
Great. So good evening, everyone. My name is Sunil Sirohi. I'm an assistant professor of pharmacology in College of Pharmacy. So my lab research focus is, if I have to just summarize everything in just one word, it is neuropsychopharmacology. So what is the meaning of that? Is neuro means we look at the neuron level. Psycho means behavior, and then once we figure out what is happening or what's the mechanism, then we tweak it with the help of some drugs. So that's basically is called pharmacology. So that is the overall crux of what we do in the lab. And in terms of research area or what exactly we do, we're interested in how binge-like eating of palatable food impacts alcohol drinking and vice versa. And you might be wondering, well, that's a very simple question. Well, actually, that seems simple, but it's very complex. Why? Because you might be, it's probably easy to really envision how many calories this food is giving to you, right? But do you know how many calories this alcohol is giving? Well, it is twice the amount, right? So the carbohydrates and protein that give you 4 calories per gram, calories per gram, but alcohol is giving you 7 calories per gram. So this is just like empty calories. It's not giving you any nutrition. And then, you might think, the adding of this together might be causing some of the metabolic disorders. So it's very unclear so far. We don't know how this happens, what's the relationship. So that's what we try to dissect in our lab. And how do we model this in our lab? So we use a rodent model. So we have rats in our lab, and we use the different diets, synthetic diets, the high fat diet or high sugar diets, and then we give the rats to eat. This is just like a treat to them. We don't give them all the time, but they get like on special days. And then we look at how does this diet impact alcohol drinking. So where they get this food and alcohol is, this is the typical rat cage. They get, so rat is in the cage, they get food and then alcohol and water to drink. And also we look at what's the motivation, like are they really motivated for alcohol? So how do we check it? This is the orange box basically. You might see that these are two levers. So rats, they press the lever to get alcohol. So if they press the lever to get alcohol, it means they are really highly motivated. And once we figure out the alcohol drinking, how does it impact, then we do a whole bunch of behavioral assays, like light and dark box, open field tratus, 22 kilohertz vocalization. So what are these? Basically, this measures the emotional status of the rat. For example, this is measuring anxiety-like behavior. This is measuring if the locomotion or the locomotor activity is impacted or not. This is measuring if I ask you that, are you sad? Well, you can tell me that you're sad, right? But how do you ask rat, are you sad? Rats can't tell you, right? So there's a way to measure that. So one method is called as 22 kilohertz ultrasonic vocalization. This is how they talk in the nature. If they are sad, they will use these vocalization. So we can monitor those and we can measure those. So after the behavior, we do a lot of biochemical and molecular assay, standard, neurotransmitter, PCR, Western blot, signaling, immunohistochemistry, and different hormone analysis. So this is just the biochemical part of assays. So now at this point, what we know is the behavior is changed. There are certain things in the body, those are changed. And then what we see is that we look at the brain level. So I remember I told you neuro. So we look at brain level, so how the brain is modulated. So what we do, we remove the rat brain, and then we remove different parts of the brain, like different parts of the brain, those are involved in these speech, mediating these effects. Then we punch these, and then once we figure out, oh, the changes are here, and then we try to infuse the drug directly in that brain area to see whether that changes the behavior in the same way or opposite. So basically, we are from top to bottom, we are looking at how or what are the neurobiological bases of the altered behavior that you see following these palatable diets. So typically in my lab, we have built students, before this pandemic, the little students would come in the lab, they will just do everything, they will do the essays, they will do analysis of the data, but due to the pandemic, basically they are just working remotely. So right now they're just analyzing data, where we have lab meetings that they talk about the data, or we discuss about data and how to move forward. And once we have project completed, we definitely have abstract all the time, and then we have publication in the end after we have enough data to really write a paper. So this is Star Villavaso, she was a former Bill student. Similia was also former Bill student. So we got publication and Bill students really equally contribute in that. So that really is a very quick spiel. I hope so. I'm really under time. I didn't take too much time, but I can talk to length, maybe 1 hour, but it's up to you if you want to listen to it. So if somebody does not ask question, there are two possibilities. One, everything is clear. Second possibility is, well, nothing is clear. I don't know what is there, but feel free to email me. My email address is right here. So this is my email address if you want to contact me. This is our lab phone, office phone. So if you want to email me or call me, anytime, I will be happy to address any questions.
Thank you so very much. We appreciate it.
Okay, bye.
Well, I would like to thank everyone for turning out today to hear about some of the fantastic research that professors at Xavier are doing. We hope that you enjoyed the presentation and it kind of helps you get to thinking about who you would like to meet with in order to set up those mentor meetings for one-on-one interactions with professors to determine which labs you would most likely like to be in. Thank you very much for coming and hope you have a good evening.
Bye-bye, everybody.
Bye, everyone. Have a good day.
Have a good day.